Hi guys, so this is a little overview of the different types of microscopes and a little bit more about how to use the microscope that we're going to be using in class. So, microscopes are instruments designed to produce a magnified visual or photographic image of objects that are too small to be seen with the naked eye. And anything you want to look at underneath a microscope can be called your specimen. So what kind of specimen you're looking at may dictate what sort of microscope you will need to use to see it. So you can see here 6 different pictures of microscopes. The first, or the top 3, top row, are all developed in about around the 1600s. They are pretty archaic, but then you can see the bottom one on the left is developed in the 1700s, and then also in the 1800s, the one in the middle. So you can see that those are not that much different than the microscopes that we are using today, the one kind of in the red. So which microscope you decide to use depends on what the specimen is that you're looking at. So there are lots of different kinds of specimens we can look at on our microscope. We can have ones that are currently alive, or there are things where we need to look at the surface, or things we need to look on the inside at high resolution or at low resolution. We want to build a 3D model from the results, or avoid any sort of removing any moisture from the sample, which might cause the sample to look different under the microscope. So based upon each of these criteria, we would choose a specific type of microscope. Here I have a link to a great website, an interactive website that allows you to decide which microscope is best for which situation you have. There are generally 3 different types of microscopes. You have the optical category, which includes the compound, stereo, or confocal laser. Then you have the electron microscopes, which include the transmission electron microscopes, the TEM, or the scanning electron microscopes, the SEM. And then finally there's the scanning probe microscopes, and we're going to use the next couple slides, we're going to talk about each of these different categories of microscopes. So we can use light, visible light, to see our items that we're going to look at, so our specimens. So these light microscopes, or optical microscopes, can make our image by refracting light with the optical lenses. The first microscopes that were invented actually belong to this category, so those ones I was talking about at the beginning. The price of an optical light microscope varies from really cheap to nearly unaffordable. So optical microscopes can be further subdivided into several categories. You've got the compound microscopes, which we're going to be using today. These are 2 lens systems. You've got the objective and the ocular eyepiece with a maximum magnification of about 1000x. Then you've got the stereo microscope, which is also called a dissecting microscope. And these microscopes magnify up to about 100x, so they just supply a 3-dimensional view of a specimen. They're used for observing like opaque objects, so things that are not transparent, just like you're learning in English class. These light microscopes let us see about 1 micron or 200 nanometers because they bounce light off surfaces to create images. So you see the picture here of the red blood cells. That's about a magnification of 400x. Just to put things into perspective, our eye can see about 20 microns, and a human hair is only about 50 to 100 microns thick, depending upon whose hair it is. So this is another light microscope, but unlike the compound and stereo microscopes, the confocal laser scanning microscopy microscope is reserved for research organizations, and it allows us to scan samples in depth. So a computer is able to assemble the data to make these really cool 3D images that you can see in this picture. They use UV light and really high resolution for that depth selectivity. In addition to using light to view our specimen, we can also use electrons to see the specimen. So electron microscopes can magnify up to 2 million times. This is possible because the wavelength of the high energy electrons is really small. But at the same time, the high energy electrons are really tough on the sample and the specimen being observed. So if we're going to be observing a biological specimen using an electron microscope, that specimen needs to be coated with a very thin layer of metal before it can be observed. This type of technology seems like it's really advanced. However, these scanning electron microscopes, or SEMs, were developed in the 1930s which means they've been in use for about 100 years. So there are two types of electron microscopes I'm going to talk to you guys about. There's actually lots of them, but the scanning electron microscopes, or the SEM, allow the electron beam to be projected on the sample. The electrons do not go through the sample, but actually bounce off. This way it's possible to visualize the surface of the structure of the specimen. So the image kind of appears three-dimensional. You can see in the top left-hand corner the image of some pollen that has been taken with an SEM. And then there's the transmission electron microscopy or microscope, the TEM. And in this case, the electron beam is passed through the sample, and the result is a two-dimensional image. And you can see the mitochondria here on the top right-hand corner that was taken by a TEM. And finally, the last way to use the microscope is to actually touch the surface of what you're visualizing. And these are called scanning probe microscopes. So scanning probe microscopes involving running a nanometer-sized probe along the surface and then measuring how much it deflects or how much it moves. And that allows a computer to kind of put together an image. So we can use really, really small things like atoms. So there are, one of the most useful ones, or well-known ones, is the atomic force microscope. So this teeny tiny tip moves up and down in response to the electromagnetic forces between the atoms on the surface and the tip that's measuring it. You can see this really cool image of an E. coli with the pila and the flagella that's been taken with an AFM. So here's an image of a compound light microscope. This is the microscope that we're going to be mainly using in class. And the one thing to note, which you guys have already labeled all of the different components of the microscope, but that the total magnification of the specimen that you're viewing is when you multiply the eyepiece lens magnification, which is 10x, by the objective lens magnification, which could be anywhere between 4x, 40x, 100x, and that will give you your total magnification. There are different types of prepared slides that we can view. You guys are going to be viewing a few in class. And these prepared slides are usually tissue that's first killed and then fixed in some sort of preserving material. It's then infiltrated and embedded in a sort of plastic called paraffin, and then the resulting block, this tissue in this plastic, is cut into really teeny sections, and then those sections are mounted onto the glass slide. So the paraffin was removed so that all that plastic is removed, and so the material is just fixed onto the slide and allows you to be able to see the light pass through the prepared slide and see the section or the specimen that you're actually looking at. So fresh mounts or wet mounts are biological material that may involve whole organisms, freehand sections cut with a razor blade or part of an organism, or even material which is mechanically or chemically teased apart. So the material is placed on a drop of water in the center of a clean slide. Some care must really be taken when you're lowering the cover slip to avoid trapping any air bubbles, so make sure you put that on at a 45-degree angle and lower it slowly. And then, since this type of preparation will actually dry up after a little while, you might want to add a little drop of water along one edge of the cover slip. Sometimes it's helpful to increase the contrast of a certain cell structure so that they can be seen more clearly. So there are certain chemicals that can dye parts of cells for these purposes, and those chemicals are called stains. And there's lots of different kinds of stains, ones that have other colors, ones that dye specific parts of the cells. And you can see here we've got human cheek cells that have been dyed with a blue stain, and in the prior slide they were not dyed, and so it's a little bit harder to see the nucleus and the edges of the cell. So when preparing a slide that has a stain or a wet mat that has a stain, it's really good to not clump your cells. This is pretty good to think about even when you're preparing any slides for a light microscope, because the more cells you have, the less light's going to be able to be attracted through and be able to be seen through that specimen. You can see in the picture for the poor prep that the stain actually only stains just a few of the cells, the ones that are on the surface and not the clumps at the bottom. And the good prep stains all the cells that you have collected for your specimen. So here's just a review of some general microscope care. Always carry with two hands, one on the base and one on the arm. always place the low power objective. So for us, I think it's a 4 when you're finished with the microscope. And you also want to always start with that low power objective when examining the slide, because the high power objective is really close to the slide, and it might even scratch or break your slide when you start to use that one first. And then when you're using a high power objective, something over 400x, only use the fine adjustment knob, not the coarse one, because if you use the coarse one, you're going to bring that slide up right into the lens, and it might break either the slide or the lens. And then when cleaning lenses of a microscope, always use lens paper. Never use your finger or any sort of other material. And don't remove any parts of the microscope. In addition, if there's any sort of malfunction with the microscope, please let Dr. Pacifica or I know. And finally, we want to keep the microscope dust-free by covering it with its protector, with its plastic cover after its use, and store it away properly in the cabinet.